Nanoparticles stimulated by microwaves to combat cancer. A physicist at the University of Texas at Arlington has proposed a new concept for treating cancer cells. In a recently published paper in the journal Nanomedicine, Nanotechnology, Biology and Medicine, UTA physics professor Wei Chen and a team of international collaborators advanced the idea of using titanium dioxide, titanium-4 oxide, nanoparticles stimulated by microwaves to trigger the death of cancer cells without damaging the normal cells around them. The method is called microwave-induced radical therapy, which the team refers to as microdynamic therapy, or MDT. The use of titanium-4 oxide nanoparticles activated by light and ultrasound in cancer treatments has been studied extensively, but this marks the first time researchers have shown that the nanoparticles can be effectively activated by microwaves for cancer cell destruction, potentially opening new doors to treatment for patients fighting the disease. Chen said the new therapy centers on reactive oxygen species, or ROS, which are a natural byproduct of the body's metabolism of oxygen. ROS help kill toxins in the body, but can also be damaging to cells if they reach a critical level. Titanium-4 oxide enters cells and produces ROS, which are able to damage plasma membranes, mitochondria and DNA, causing cell death. Cancer cells are characterized by a higher steady-state saturation of ROS than normal, healthy cells, Chen said. This new therapy allows us to exploit that by raising the saturation of ROS in cancer cells to a critical level that triggers cell death without pushing the normal cells to that same threshold. The pilot study for this new treatment concept builds upon Chen's expertise in the use of nanoparticles to combat cancer. Chen's collaborators hail from the Guangdong Academy of Medical Sciences and Beihang University. The team conducted experiments that demonstrate the nanoparticles can significantly suppress the growth of osteosarcomas under microwave irradiation. While titanium-4 oxide and low-power microwave irradiation alone did not effectively kill cancer cells, the combination of the two proved successful in creating a toxic effect for the tumor cells. Microwave ablation therapy has already proven to be an effective treatment against bone cancer, obtaining better results than MDT. However, MDT has applications for combating other types of cancer, not just the osteosarcomas used for this pilot case. Using light to activate ROS, as is seen in photodynamic therapy, can be challenging for the treatment of tumors deeply located within the body. In contrast, microwaves lend the ability to create deeper penetration that propagates through all types of tissues and non-metallic materials. This new discovery is exciting because it potentially creates new avenues for treating cancer patients without causing debilitating side effects, Chen said. This targeted, localized method allows us to keep healthy cells intact so patients are better equipped to battle the disease. The results of the pilot study indicate MDT as a promising approach for cancer treatment even though the method is still being developed and its limitations explored. The research team has filed a patent for MDT. Next, they plan to turn their attention to understanding the physics and internal mechanisms behind the powerful combination of microwaves and titanium-4 oxide. Dr. Chen continues to build a research portfolio that holds transformative implications for cancer treatment, said Alex Weiss, chair of UTA's Department of Physics. This new work is exemplary of the spirit of discovery we strive to embody at UTA. I look forward to what Dr. Chen and his collaborators can accomplish in the coming phases of this research as they pioneer a potential new avenue for combating cancer. Zebrafish capture a window on the cancer process. Cancer-related inflammation impacts significantly on cancer development and progression. New research has observed in zebrafish, for the first time, that inflammatory cells use weak spots or microperforations in the extracellular matrix barrier layer to access skin cancer cells. The research, led by the University of Bristol and published in Cell Reports Today Tuesday 4 June, used translucent zebrafish to model several sorts of skin cancer and live image how inflammatory cells find the growing cancer cells in the skin. To access the cancer cells, the immune cells need to first breach an extracellular matrix barrier layer called the basement membrane zone. The researchers observed weak spots in the basement membrane zone, which the inflammatory cells use as easy routes to access the cancer. Those clones of cancer cells nearest to the weak spots tend to receive more inflammatory cell visits and as a consequence they grow faster. Paul Martin, professor of cell biology in the schools of biochemistry and physiology, pharmacology and neuroscience at the University of Bristol, said, "As the zebrafish are translucent, we can watch inflammatory cells interacting with cancer cells in ways not possible in our own tissues. This window on the cancer process has revealed weak spots in the barrier layer that inflammatory cells must breach in order to access and feed the cancer cells within the skin. Now we know these microperforations exist, we can target them with cancer therapeutics." Inflammatory cells, white blood cells, are known to be key players encouraging cancer malignancy from the earliest stages of cancer initiation, but it has been unclear how they gain access to the cancer cells, which generally develop within epithelial tissues, which have a barrier layer of extracellular matrix between them and the tissues from where the inflammatory cells arise. Inflammatory cells could kill the growing cancer but instead tend to nourish it and encourage cancer progression. The study's findings have clear clinical relevance to cancer patients because microperforations in the basement membrane zone have been shown to occur in human airways and guts, and so could act as similar portals to let inflammatory cells gain access to the cancer. The research has been funded by CRUK and Wellcome Trust. Killing the unkillable cancer cells. Why do some cancer cells evade therapy? To understand this problem, we need to look at the molecular machinery inside the cancer cells that regulates their ability to grow, stay alive, and divide, producing more cells. Scientists have identified many mechanisms within this machinery, and they have designed drugs and other treatments that interfere with them, blocking their ability to divide, or even killing them. Clearly, these treatments are not able to kill all the cells within a tumor. This must mean that some cancer cells can use alternative mechanisms which are not affected by current treatments. In this way, they evade therapy and regenerate the tumor, leading to recurrence. The lab of Andreas Andrautselis Theotokos at the Department of Internal Medicine 3 (MK3) at the Technische Universität Dresden, in Germany had previously discovered a new molecular mechanism that regulates regeneration of various tissues after injury. They named it the STAT3-SR, Hes3 signaling axis, after two of its key components, and they demonstrated that it is very important in activating stem cells that live within our tissues so that they may help the tissue regenerate in models of Parkinson's disease, ischemic stroke, multiple sclerosis, and types 1 and 2 diabetes. They posited that the same mechanism could be hijacked by some cancer cells, perhaps the elusive cancer stem cells, as a means of evading current therapies and regenerating the tumor. Research fellow Steve Poser and colleagues set to test the idea using cells from multiple patients with aggressive brain cancer. They demonstrated that, indeed, these cells can switch over from using the more established molecular mechanisms to using this new mechanism. Depending on which mechanism they were using, they were vulnerable to different treatments. The team established an international and interdisciplinary collaborative project to characterize this new cancer cell state in terms of what genes are turned on and off, what mechanical properties are affected. These are important in metastasis because they regulate how cells move and spread within the tissue, and to find treatments that kill the cells specifically when this new molecular mechanism is in use. In this way, they could block this escape route of the cancer cells. The findings are published in the Journal of the Federation of American Societies for Experimental Biology. They identified several treatments that kill the cancer cells when they operate this new mechanism. In fact, several treatments are so specific that they kill the cells only when they operate the new mechanism and have minimal effects when they operate the more established, classical cancer growth mechanisms. Many of the treatments are currently approved for use in different indications, not cancer, and so they could potentially be repurposed in oncology. Other treatments, such as the direct targeting of the gene Hes3 by RNA interference methods, would be new to man. To expedite the possibility to bring these new potential therapies to the clinic, the Technische Universität Dresden, with the support of the Transcampus Initiative, a formal collaboration between the Technische Universität Dresden, in Germany and King's College, London, UK, and the Transliterator, has formed a spin-off venture, Innate Repair. Whereas the initial focus is on aggressive brain cancer, there already is ample evidence that the same mechanism operates in many additional types of cancer. The work provides a new logic and method to identify treatments that kill these hard-to-kill cancer cells. Lymphoma trial finds combination targeted therapy effective prior to chemotherapy. Results of a Phase 2 clinical trial conducted at the University of Texas MD Anderson Cancer Center revealed that combination targeted therapy, consisting of rituximab, lenalidomide and ibrutinib (RLI), had an 84.6% overall response rate and 38.5% complete response rate (CRR) when given prior to any chemotherapy for newly diagnosed patients with a specific type of diffuse large B-cell lymphoma (DLBCL). The first-of-its-kind study examined a treatment regimen without chemotherapy for patients with non-germinal center (non-GCB) DLBCL, and, while confirmatory trials are needed, the findings suggest that patients who respond to targeted therapy initially may not need chemotherapy, currently the standard of care. The results of the trial will be shared today in an oral presentation at the 2019 American Society of Clinical Oncology annual meeting by Principal Investigator Jason Weston, MD, Assistant Professor of Lymphoma and Myeloma. The responses we've seen have been remarkable. More than 80% of our patients have responded and around 40% have had a complete response, showing no evidence of cancer, prior to receiving any chemotherapy, said Weston. All patients have gone on to receive standard chemotherapy in combination with these targeted treatments per the protocol, and, so far, we've had a 100% response rate. Large cell lymphomas, which include DLBCL, are diagnosed in approximately 30,000 individuals each year in the U.S., and non-GCB accounts for an estimated 30 to 40% of those cases. Standard treatment for large cell lymphomas is chemotherapy, but this subtype doesn't respond as well, reaching an estimated cure rate of just 50 to 60%, explained Weston. The clinical trial enrolled 60 patients at MD Anderson with non-GCB DLBCL and treated those patients with 2 cycles of RLI, followed by 6 cycles of RLI with chemotherapy. Weston's team designed the trial to bring new treatment options to these patients based on promising findings in the lab. We called the trial Smart Start because we thought this was a smarter way to start therapy for these patients, said Weston. Standard treatment for large cell lymphoma has been largely stagnant for the better part of 40 years, despite many advances in our understanding of the disease and a host of new medications. It's exciting to see an idea that worked in the lab now beginning to yield results and show this as a potentially new way forward to fight this disease. More than 90% of patients on this trial remain in remission after 1 year, said Weston. Additionally, side effects on the trial have been mild, with most driven by the chemotherapy treatment. Going forward, Weston and colleagues plan to launch clinical trials to investigate whether patients who respond well to RLI treatment upfront can receive little or no chemotherapy and still attain long-term remission.